You are listening to Life Science Connect Radio. Broadcasting live from New York and direct from the show floor of Interphex. Advancing the processing of life-enhancing drugs. Life Science Connect and Interphex, together in the Big Apple. And now, here are your hosts Todd Schnick and Todd Youngblood.
Good afternoon. This is Todd and Todd live in New York, Life Science Connect Radio on location, direct from Interphex on day 2. Todd, an exciting guest up next, but just having a great day here. We're just learning so much, interacting with some cool companies.
Your brain getting full yet?
It's darn near full.
I think I got maybe a quart left to go.
Well, we have a great guest up. Say hello to Jack Giese. He is the Director of Commissioning and Validation at Finesse Solutions. Jack, welcome to the show.
Thank you very much for having me today.
It's our pleasure, Jack. Thanks for stopping by and joining us. Before we get into a conversation around Finesse, take a few quick seconds. Tell us a little bit about you and your background.
I've been in the biotech industry now for approximately 30 years, mostly working in the manufacturing pharmaceutical medicines, design, construction of pharmaceutical facilities.
All right, well, give us the 10,000-foot view of Finesse. What do you guys do? How do you serve your market?
Our main focus is disposable transmitters and measuring devices, as well as our automation control platform. We are able to integrate with most customers' pharmaceutical disposable equipment and integrate that into the working system in their facilities.
Boy, it's interesting. We talked a lot about single use. When I hear something like disposable transmitters, are we getting to the point where everything's going to be single use in this industry?
We're trying anything to get away from cleaning and steaming validation to save that cost association. That's what we're looking for now in the industry.
Well, what's driving all that?
It's just a lot of testing, and the cost associated with doing that cleaning and steaming validation testing can run up to 10 to 20% of your project cost. So by bringing that equation back down, you could actually invest that back into better controls and technology for growing your products.
Well, walk through some of the critical products in your portfolio.
I would say the one with our flagship is our optical DO and pH sensors. These actually have a disposable sheath that's in contact with the product, and the sensor itself is removed from the process. So it could be gamma radiated with the bag, thrown away at the end of the process, and you still have the major instrument there and reusable for your next batch.
Let's back up a little bit and talk about the challenges that the industry is facing.
I think the need is to be quick and nimble and flexible, because the costs associated with bringing products to the market now are getting so large that you have to be able to make a phase 1, phase 2 clinical on a smaller scale and be able to scale it very quickly to get to the market so you can get that market share.
Is it fair to assume that Finesse is equipped to do that nimbleness?
Yes, we're a very small, and we work well with all of our customers. We mostly can integrate 5 to 6 different vendors worth of equipment, so if you're using a disposable bioreactor, it doesn't matter to our control platforms what the vendor type is. We have very quickly, within 5 minutes, be able to switch from one vendor type to another for the disposable bags.
Talk more about the integration and how important that is. I mean, there's so many different moving parts.
I think what we bring to the market with our system, since we can standardize this for a control platform, that the validation of the system is very quick and easy. We have our own validation documents will go along with the system and can make it a very affordable option for choosing our control system.
How do you simplify tech transfer? Walk us through that.
With our system, you actually have the ability to load and save programs and your files, all your file data. So if you're producing at your clinical facility, an R&D facility in one state, and need to move it across the ocean to a facility in Europe, you can actually transfer those recipes, load them onto your operating system in the new facility, and you're actually up and running with the same parameters. It's not that paper tech transfer that makes sure you're entering all those parameters correctly, when you can have hundreds and hundreds of different parameters actually to enter back into these systems.
Jack, you've talked about the speed of getting a line up and running. You've talked about cost reductions on a qualitative sense. How much money and how much time is really involved here? How much are we saving?
Typically on a stainless steel facility in the past where I've worked, can be anywhere from a 5 to 6 year project to go through the design, construction, and start up of those facilities. With a disposable facilities, we're looking at being able to do that same type of project in probably 12 to 18 month time period. So just those extra 2.5, 3 years to get the product first could mean a big difference to a company.
Well, you're exhibiting here at Interphex. What are you showcasing? What are your goals and objectives?
We have several new product lines out here, so we're trying to highlight those, more of our disposable technology. So we now have a disposable 3-liter vessel. So with our optical readers inside of that, it gives you the ability to use it once, dispose of that vessel. No more of the costs associated with cleaning, steaming, and autoclaving of a glass vessel.
What next? Let's look out 3 years, 5 years. What kind of technology do you see becoming important? And how will that impact what you're doing?
I think what we're driving towards and what the customers are asking for now is a complete package solution to a disposable facility. Flex factory, as it's being called these days. So the ability to bring in and integrate all of the upstream and downstream processing needs, bring them all together under one automation platform, collect that information so you can quickly analyze your data and release your batches.
All right, Todd, we're starting to hear about this Flex factory a lot more now.
Yeah.
Walk me through, Jack, what the value is to the end user, the customer down at the end of the line.
I would have to say that, again, being quickly to get your product back to the market. By having this, the cell culture side, the upstream processing side, titers have gone up very quickly and able to produce more with less, so smaller vessels, smaller footprints. The downstream is still trying to catch up with that technology. You're still running fairly large on your purification and recovery system. So it's trying to minimize the batch sizes on one side to meet your process on the back end.
Jack, how long do you think it'll be until the Flex factory is a common thing?
I would say that's probably 1 to 2 years out.
That's
really getting a lot of traction these days.
Holy smokes, I wouldn't have expected that. That's going to change everything. I mean, the conversations we'll have on this show in a year or two time is going to be dramatic as a result of that. I mean, it's that profound to the industry, right?
Correct.
All right.
Well, I guess, and you know how long I've been around manufacturing my whole career. Flex factory is the term I hear, but in my brain I'm always saying it's a disposable factory. And it just blows my mind that we're even talking about it, no less doing it in a year or two.
Yeah, pretty fascinating. Well, Jack, I hate to say it, but we're about out of time. Before we let you go, how can people get in touch with you, and where can they learn more about Finesse Solutions?
You can come to our website, www.finesse.com, and you can see all of our new instruments and products available on the market today. Contact information is there for our sales force also.
All right. Jack Giese, Director of Commissioning and Validation with Finesse Solutions. Jack, it was great to have you. Thanks for stopping by and joining us.
Thanks a lot, Todd.
All right. Well, that wraps this broadcast. On behalf of our guest, Jack Giese, my co-host, Todd Youngblood, I'm Todd Schnick, Life Science Connect Radio's live coverage of Interphex. We'll be right back.